Bianca Dang, and today I'll be presenting about how ex-vivo focused ultrasound sonication can potentially induce histologic changes in neural tissue. Ongoing projects in our lab have patients with temporal lobe epilepsy treating it with focused ultrasound at intensities of 5,760 milliwatts per centimeter squared. Several days after sonication, the sonicated brain regions are surgically resected and the tissue is examined. The skull is known to attenuate up to 90% of the transmitted energy, so unsurprisingly, there was no detectable damage in these resected tissues. However, in order to know whether the intensity levels were safe or not, we had to determine this through a series of experiments in which we attempted to induce thermal and mechanical injury to the brain cells of the human cadaver via ultrasound pulsation sonication. We conducted our study on 7 brain samples that were taken from cadaver temporal lobes. Each sample was sonicated for 60 seconds at either 0, 750, 1200, 1500, 3000, 6000, or 12,000 milliwatts per centimeter squared isepta. The figure on the right here, shows the experimental setup, with the petri dish in the position of the focus. After sonication, tissue was placed in formalin for 48 hours, then embedded in paraffin wax, sliced at 4 microns, and stained using hematoxylin and eosin. For each block, we kept one piece not sonicated, but otherwise it went through all of the same exact processing steps in order to serve as a control piece. After all of this, we had a blinded investigator use light microscopy in order to analyze the neural tissue samples as well as conduct cell cross-sectional areas in which were later converted into volumes using geometric relationships. Here, you can see the distribution of the cell volumes at the different stimulation intensities. We found that regardless of the source, all of the tissue that was sonicated below 25 watts per centimeter squared was visually indistinguishable from the control pieces. Sonication above 25 watts per centimeter squared demonstrated irreversible damage to the cell's integrity, evidenced by spongiosis and blood vessel destruction on light microscopy. Side by side, the neural tissue sonicated, again, is indistinguishable from the control piece. Here we have a sonicated piece and a control piece. As you can see, there is visually no differences. On the left, we have the sonicated piece at 1200 milliwatts per centimeter squared, and on the right, we have the control without the sonication. We also conducted various statistical analyses to confirm this, and had statistically significant results. The takeaways here from our study is that focused ultrasound does not cause significant damage to cadaver neurons, and it is likely safe for human clinical trials. However, it is notable to mention that the tissues did not have a piece of skull in the way to attenuate some of the ultrasound energy. As such, an in vivo approach would require roughly 250 watts per centimeter squared intensity in order to have a similar impact on the tissue. However, even disregarding this, at 25 watts centimeter squared, there is a great potential that the 90% attenuation by the skull will result in significant heating, leading to burns on the skin potentially. For this reason, further safety testing, of course, is needed to assess the degree of heating in the skin at these intensity levels prior to commencing human subject work. The device used was made by BrainSonics Corporation, and we'd like to also acknowledge the Pacific Institute of Medical Research, as well as the Gerald J. and Dorothy R. Friedman New York Foundation for Medical Research. And of course, I would personally like to thank Norman M. Spivak, as well as our co-authors and collaborators. Thank you so much for your time, and have a wonderful day.